And so this morning, I just want to, before we go on to influenza, I just want to finish a couple of things from the genetics lecture, because again, there are a couple of terms that I want to introduce and concepts that I want to introduce. So we talked about mutation, recombinations, and how the viruses are affected by these things, and how the nature of the genome, whether they're DNA or RNA, whether they're segmented or non-segmented, affects their genetics. And I just wanted to pick up a couple of other things before we go off into the more detailed clinical stuff, because some of this comes back to haunt us again. So one thing is the recombination is exchange of genetic material at the nucleic acid level, but viruses can cooperate at the functional level by exchanging proteins, which is complementation. And just to show an example of this, supposing we were to infect a cell with 2 rhabdoviruses, and that this one had got a temperature-sensitive N protein, nucleic capsid protein, and as I said last time I lectured, yesterday, seems a long time ago, but yesterday. Anyway, the temperature-sensitive proteins are frequently undergo some kind of conformational change at higher temperature, such that they no longer carry out their original functions. So we could grow this virus at low temperature, but we can't grow it at higher temperature because the nucleic capsid protein won't function at higher temperature. We infect with another virus, the blue virus, same rhabdovirus again, but it's got a temperature-sensitive maturation protein, so it won't grow at the higher temperature. If we infect them both into a cell, though, at the higher temperature, they will be able to grow because this one will make enough nucleic acid of the correct maturation protein to provide it for this, so this one can be packaged, and ditto for the N protein. So between them, they can come up with some good M protein and some good N protein, and they can get cooperation at the protein level. There's no change in the genomes. So though neither of these viruses could grow alone at the higher temperature, together they can grow in the cell. But if you look at the progeny virus will have the other virus's N or M protein in its internal parts here. So it will have those proteins in the virion particle, but the nucleic capsid will be the original the blue genome will contain the original blue genome or the original pink genome, and so they will breed true. It will get a virus particle which has got proteins from the other one, but if you grow it in new cells, it will behave just as if it was the original virus that you had here, because there's no genetic change. So complementation is a way that some viruses can help each other out on a temporary basis. It's difficult to know whether this ever happens, because if it happens in just one cell in your body and one virus helps another virus just temporarily, then it can lead to events which can eventually cause disease, but it's going to occur in a very rare number of people, and there's no way you ever know what the basis is. But we suspect that occasionally complementation can happen, and I might come back to another mention of where that might be. So, and another point is that because they're exchanging proteins, usually the things that complement are because the viruses have got mutations in different genes. It's rare for 2 bad copies of the same protein to be able to complement each other. But a kind of complementation that we do see is something called defective viruses. And defective viruses are viruses which lack some gene or some part of some gene or other, and in lacking that gene, they can't undergo a complete replication cycle because they're missing something that's important. So if another virus comes in and provides that gene, that protein rather, the product of that gene, that defective virus can replicate because it's using proteins made for it by the incoming virus, the complementing virus. So in terms of defective viruses, these incoming viruses which provide the missing proteins are called helper viruses. So what how defective can a virus be? A virus can actually be extremely defective. So you can have viruses in which basically the genome only contains the two signals that are essential for that virus. And the one signal says, package me, and the other signal says, replicate me. So as long as they've got the right signal for replication and the right signal for packaging, they can actually lose everything else if it has a helper virus to provide everything else. These are two things that have to be on the genome. These signals are not diffusible. They're part of the genome. But for making proteins, if you delete the genes that make proteins, then if another virus makes the proteins, they can diffuse over and help this genome be copied, packaged, and basically produce new particles. So this is your smallest defective particle that you can get. So for some examples of defective viruses, you're going to be hearing them later on. Some retroviruses are defective. They have some of them pick up host cell genes, and in doing so, they actually lose a bit of their genome because it seems that they don't have room to package the full genome. So if they pick up something, they have to give up something in return. So they use helpers, and frequently the helper is actually the parental virus, as it were, for the retrovirus that has lost a gene, but frequently has picked up a host cell gene. So my husband will be talking more about those. We've already talked about hepatitis delta virus, which is this weird thing that has got one foot in the virus camp and one foot in the viroid camp. And I've already said that it doesn't encode a envelope attachment protein, and it uses an unrelated helper to package itself, and that is hepatitis B virus. So it uses the hepatitis B virus envelope to package itself. So that is an unrelated helper, but again, it's complementation because the hepatitis B virus is providing something for hepatitis delta virus to replicate. So these are two examples of viruses which use other viruses to provide these helper functions. So these viruses are defective in some way or other, but some of these defective viruses are also called defective interfering viruses. And why the word interfering? Well, if you're a defective virus, you're competing with the helper virus because you're using helper virus functions. And that competition can be so severe that you actually decrease the replication of the helper virus, partly because these things tend to have pretty small genomes. They're frequently smaller than the helper virus. They replicate so quickly that they get ahead and make lots and lots of copies, and the helper virus is actually somewhat slower because it's got a bigger genome to copy. And so they compete very, very strongly sometimes for the products that the helper virus is making, and that has a negative effect on the helper virus. So in that case, they actually interfere with the production of the helper virus. So if defective interfering particles arise in a cell, they can actually suppress the replication of the helper virus. And this can be, sometimes it might be good news, you might lower the replication of the helper virus. Sometimes it can be bad news, because if the helper virus, if it replicated properly, would have caused a lytic infection and killed the cell, then if you suppress it such that it doesn't kill the cell, you help it to establish persistent infections. So these may modulate the wild-type infection in one way or another, and actually we do have naturally occurring defective interfering infections in humans, and they're associated, defective interfering measles virus particles are associated with a disease called subacute sclerosing panencephalitis, which is a long, slow, fatal disease that can develop as a complication of measles. So this disease, one of the things about it is it took a while to realize what was causing it, because if you look in the brain of affected individuals, the brain has got these defective interfering particles in. But if you try and isolate virus from the brain, you won't be able to do it, because the wild type does not seem to, is either not around or is thoroughly suppressed. And so the defective interfering particles, you can't grow them out in culture. So you don't realize that you've got the virus there, but you do. And these defective interfering particles can replicate because they're actually defective in putting together their cell envelopes. But if you remember, measles is a fusion virus. So they can actually replicate, make new copies of the nuclear capsid. They just can't package and get out of the cell, but they can fuse with the next cell. So this sort of infection can creep by forming syncytia gradually, which is probably part of the reason it's such a slow infection. But eventually, it takes over. So one thing to remember with defective interfering particles is you may not be able to isolate the virus from the infected material, but that doesn't mean it's not there. So if you add a helper virus, you can then show that this material is there. So that's another example of defective interfering particles, which are themselves examples of some kind of complementation. If they're going to produce if they're going to get out and be able to infect new cells, they're going to need a helper. Another example of a kind of complementation is something which is called phenotypic mixing. So here, if we take two viruses, this could, for example, be poliovirus, and this could be Coxsackie virus, which is another member of the same family. If you look at them, they have their own phenotypic properties, and so if you look at the outside, it will have the typical surface antigens of a poliovirus or of a Coxsackie virus. And if you were to grow them up, this would have particular properties that you'd associate with a Coxsackie virus or with a poliovirus. But you can actually grow these in the same cell, and if they grow in the same cell, although they've got their own individual nuclear capsid proteins, they can actually incorporate some of the other person's or the virus's nuclear capsid proteins into their particles. So you can get particles that come out that are mixed in terms of the surface proteins. The genome will either be a Coxsackie or a polio genome, but if you grow them in the same cell, they can be packaged in a nuclear capsid, which is actually temporarily a mixed nuclear capsid, because you can get proteins, these proteins are so alike, they can actually form part of the same nuclear capsid. This means that if you look at this from an antigenic point of view, as far as the host is concerned, the surface looks like it's part Coxsackie and part polio. So if you've got antibodies against polio, you can neutralize the polio attachment proteins, but you can't neutralize the Coxsackie attachment proteins, and vice versa. If you've had Coxsackie but don't have antibodies against polio, you can neutralize the the green proteins, but not the purple proteins. So to really neutralize these virus particles, you're going to need to have antibodies against both attachment proteins. So temporarily, this virus has acquired the ability to infect the cells that the green virus infects, and this virus, the green virus, has acquired the ability to infect the cells that the purple virus infects. And they've also acquired the ability to escape your immune system if you've only got immunity to one or other of them. So this can enable them temporarily to get into places where they wouldn't otherwise go. And again, that may cause some of the rare complications one can see in viruses. So again, there's no changes in the genome, but you can alter the host range, and they become resistant to antibody neutralization. Now, you can get an extreme example of this in certain circumstances, and in which the green virus gets entirely packaged with purple proteins, and the purple virus gets entirely packaged with green proteins. So temporarily, you've got a virus whose inside is devoted to being a polio virus, but whose outside is devoted to being a Coxsackie virus. You actually don't so frequently see this. You see this more frequently in the envelope viruses, which are much more sloppy about what they will accept as an outer surface. The naked nuclear capsid viruses, they usually have to be pretty related to exchange nuclear capsid proteins. But the envelope viruses will happily accept envelopes which have got the surface proteins of other envelope viruses in them. So this virus is an example of phenotypic mixing, but sometimes these extreme virus examples are called pseudotypes because they're fake, they're pseudo. They look as if they're purple, but they're really green inside, and they will breed true to whatever was the nucleic acid that was packaged in them. Are there any questions there? Okay. So let's switch over to influenza.